Acid-base homeostasis, Wikipedia article audio. Acid-base homeostasis is the homeostatic regulation of the pH of the body's extracellular fluid. The proper balance between the acids and bases in the ECF is crucial for the normal physiology of the body, and cellular metabolism. The pH of the intracellular fluid and the extracellular fluid need to be maintained at a constant level. Many extracellular proteins such as the plasma proteins and membrane proteins of the body's cells are very sensitive for their three-dimensional structures to the extracellular pH. Stringent mechanisms therefore exist to maintain the pH within very narrow limits. Outside the acceptable range of pH, proteins are denatured, causing enzymes and ion channels to malfunction. Acid-base balance. Imbalance. In humans and many other animals, acid-base homeostasis is maintained by multiple mechanisms involved in three lines of defense. The first line of defense are the various chemical buffers which minimize pH changes that would otherwise occur in their absence. They do not correct pH deviations, but only serve to reduce the extent of the change that would otherwise occur. These buffers include the bicarbonate buffer system, the phosphate buffer system, and the protein buffer system. Physiological corrective measures make up of the second and third lines of defense. This is because they operate by making changes to the buffers, each of which consists of two components, a weak acid and its conjugate base. It is the ratio concentrations of the weak acid to its conjugate base that determines the pH of the solution. Thus, by manipulating firstly the concentration of the weak acid, and secondly that of its conjugate base, the pH of the extracellular fluid can be adjusted very accurately to the correct value. The bicarbonate buffer, consisting of a mixture of carbonic acid and a bicarbonate salt in solution, is the most abundant buffer in the extracellular fluid, and it is also the buffer whose acid-to-base ratio can be changed very easily and rapidly. Thus the second line of defense of the pH of the ECF consists of controlling of the carbonic acid concentration in the ECF. This is achieved by changes in the rate and depth of breathing, which blows off or retains carbon dioxide in the blood plasma. The third line of defense is the renal system, which can add or remove bicarbonate ions to or from the ECF. The bicarbonate is derived from metabolic carbon dioxide, which is enzymatically converted to carbonic acid in the renal tubular cells. The carbonic acid spontaneously dissociates into hydrogen ions and bicarbonate ions. When the pH in the ECF tends to fall, the hydrogen ions are excreted into the urine, while the bicarbonate ions are secreted into the blood plasma, causing the plasma pH to rise. The converse happens if the pH in the ECF tends to rise. The bicarbonate ions are then excreted into the urine and the hydrogen ions into the blood plasma. An acid-base imbalance is known as acidemia when the acidity is high, or alkalemia when the acidity is low. The pH of the extracellular fluid, including the blood plasma, is normally tightly regulated between 7.32 and 7.42, by the chemical buffer S, the respiratory system, and the renal system. Aqueous buffer solutions will react with strong acids or strong bases by absorbing excess hydrogen H+ ions, or hydroxide O ions, replacing the strong acids and bases with weak acids and weak bases. This has the effect of damping the effect of pH changes, or reducing the pH change that would otherwise have occurred. But buffers cannot correct abnormal pH levels in a solution, be that solution in a test tube or in the extracellular fluid. Buffers typically consist of a pair of compounds in solution, one of which is a weak acid and the other a weak base. The most abundant buffer in the ECF consists of a solution of carbonic acid, and the bicarbonate salt of, usually, sodium. Thus, when there is an excess of O ions in the solution, carbonic acid partially neutralizes them by forming H2O and bicarbonate ions. Similarly, an excess of H and ions is partially neutralized by the bicarbonate component of the buffer solution to form carbonic acid, which, because it is a weak acid, remains largely in the undissociated form, releasing far fewer H and ions into the solution than the original strong acid would have done. The pH of a buffer solution depends solely on the ratio of the molar concentrations of the weak acid to the weak base. The higher the concentration of the weak acid in the solution, the lower the resulting pH of the solution. Similarly, if the weak base predominates, the higher the resulting pH. This principle is exploited to regulate the pH of the extracellular fluids. For the carbonic acid bicarbonate buffer, a molar ratio of weak acid to weak base of 120 produces a pH of 7.4, and vice versa. When the pH of the extracellular fluids is 7.4, then the ratio of carbonic acid to bicarbonate ions in that fluid is 120. This relationship is described mathematically by the Henderson-Hasselbalch equation, which, when applied to the carbonic acid bicarbonate buffer system in the extracellular fluids, states that the pH of the extracellular fluids can thus be controlled by separately regulating the partial pressure of carbon dioxide, and the bicarbonate ion concentration in the extracellular fluids. There are therefore at least two homeostatic negative feedback systems responsible for the regulation of the plasma pH. The first is the homeostatic control of the blood partial pressure of carbon dioxide, which determines the carbonic acid concentration in the plasma, and can change the pH of the arterial plasma within a few seconds. The partial pressure of carbon dioxide in the arterial blood is monitored by the central chemoreceptors of the medulla oblongata, and so are part of the central nervous system. These chemoreceptors are sensitive to the pH and levels of carbon dioxide in the cerebrospinal fluid. These chemoreceptors are sensitive primarily to changes in the partial pressure of oxygen in the arterial blood and are therefore not directly involved with pH homeostasis. The central chemoreceptors send their information to the respiratory centers in the medulla oblongata and pons of the brainstem. The respiratory centers then determine the average rate of ventilation of the alveoli of the lungs, to keep the partial pressure carbon dioxide in the arterial blood constant. The respiratory center does so via motor neurons which activate the muscles of respiration. A rise in the partial pressure of carbon dioxide in the arterial blood plasma above 5.3 kPa reflexly causes an increase in the rate and depth of breathing. Normal breathing is resumed when the partial pressure of carbon dioxide has returned to 5.3 kPa. The converse happens if the partial pressure of carbon dioxide falls below the normal range. Breathing may be temporarily halted, or slowed down to allow carbon dioxide to accumulate once more in the lungs and arterial blood. The sensor for the plasma HCO(3) concentration is not known for certain. It is very probable that the renal tubular cells of the distal convoluted tubules are themselves sensitive to the pH of the plasma. The metabolism of these cells produces CO2, which is rapidly converted to H and HCO(3) through the action of carbonic anhydrase. When the extracellular fluids tend towards acidity, the renal tubular cells secrete the H and ions into the tubular fluid from where they exit the body via the urine. The HCO3 ions are simultaneously secreted into the blood plasma, thus raising the bicarbonate ion concentration in the plasma, lowering the carbonic acid/bicarbonate ion ratio, and consequently raising the pH of the plasma. The converse happens when the plasma pH rises above normal. Bicarbonate ions are excreted into the urine, and hydrogen ions into the plasma. These combine with the bicarbonate ions in the plasma to form carbonic acid, thus raising the carbonic acid-bicarbonate ratio in the extracellular fluids, and returning its pH to normal. In general, metabolism produces more waste acids than bases. The urine is therefore generally acid. This urinary acidity is, to a certain extent, neutralized by the ammonia which is excreted into the urine when glutamate and glutamine are deaminated by the distal renal tubular epithelial cells. Thus some of the acid content of the urine resides in the resulting ammonium ion content of the urine, though this has no effect on pH homeostasis of the extracellular fluids. Acid-base imbalance occurs when a significant insult causes the blood pH to shift out of the normal range. An abnormally low pH in the ECF is called an acidemia, and an abnormally high pH is called an alkalemia. A second pair of terms is used in acid-base pathophysiology: acidosis and alkalosis. They are often used as synonyms for acidemia and alkalemia, though this can cause confusion. Acidemia refers unambiguously to the actual change in the pH of the ECF, whereas acidosis, strictly speaking, refers to either a rise in the amount of carbonic acid in the ECF or to a decrease in the amount of HCO3 in the ECF. Either change would on its own cause an acidemia. Similarly, an alkalosis refers to a rise in the concentration of bicarbonate in the ECF, or to a fall on the partial pressure of carbon dioxide, either of which would on their own raise the pH of the ECF above the normal value. The terms acidosis and alkalosis should always be qualified by an adjective to indicate the cause of the disturbance: respiratory, or metabolic. There are therefore 4 different acid-base problems: metabolic acidosis, respiratory acidosis, metabolic alkalosis, and respiratory alkalosis. One or a combination these conditions may occur simultaneously. For instance, a metabolic acidosis is almost always partially compensated by a respiratory alkalosis, or a respiratory acidosis can be completely or partially corrected by a metabolic alkalosis. Whether an acidosis causes an acidemia or not depends on the magnitude of the accompanying alkalosis. If the one cancels the other out, then there is neither an acidemia or an alkalemia. If the accompanying alkalosis overwhelms the acidosis, then an alkalemia results, whereas if the acidosis is greater than the alkalosis, then an acidemia is the inevitable result. The same considerations determine whether an alkalosis results in an alkalemia or not. The normal pH in the fetus differs from that in the adult. In the fetus, the pH in the umbilical vein pH is normally 7.25 to 7.45, and that in the umbilical artery is normally 7.18 to 7.38. Body water, intracellular fluid/cytosol. Extracellular fluid.